Okay, so this is video number 2, and it's going to focus on osmosis and diffusion. What is osmosis and what is diffusion, and what are the differences between both of them? That's something I'm going to be trying to explain by the end of this video. But the first thing I want to talk about is description of diffusion. And diffusion, like I explained to you guys in class, and you guys actually observed, is the movement of molecules, just of anything, basically from a place where there's so much of it to a place where there's less of it, less amount of it. So if someone sprays a perfume in a certain environment, a certain area, at some point the smell of the perfume is going to, or the fragrance of the perfume is going to spread across a room or spread just around the container or wherever that place or where the person is staying, or wherever it was sprayed. So that's diffusion. Diffusion is movement of molecules from a region of high concentration to a low concentration. And we're going to see how osmosis is similar and also very different from diffusion. I've copied most of the materials on this slide from the actual slide that was shared with everyone in class, that we actually used to present in class. So you can always go back to reference those materials. Something I wanted to talk about on this slide is the difference between glucose and starch, and why that's important when we talk about movement of molecules through a semi-permeable membrane. Now, by now, you guys know what semi-permeable membrane means, right? Permeable means that something can go through it, and semi-permeable means that some things can go through it and some things cannot, right? Impermeable means that it doesn't allow anything to go through it. So in this slide, I want to talk about the difference between glucose and starch. And as you can see from this picture, starch has so many more single glucose, right? If we want to put it in that term, or I can say that glucose is just one of one simple single molecule, and starch is actually multiple of this same glucose, right? So you have many more of this glucose, and what would you expect would be the result in terms of size, right? So if you guessed that glucose would be smaller than starch, you guessed it right. And that's something that's going to affect us when we look at diffusion of molecules through a semi-permeable membrane. So something that you need to understand is that diffusion happens through membranes, and they happen with a factor that affects diffusion is actually size. So in terms of a pore or a gap, if we were to pass glucose through a certain gap, or starch, which one do you think would find it easier to move through a certain gap, or will be able to permeate through a certain size? Of course, you would think it's glucose, and that's going to be correct. And the reason why is because glucose is smaller in size, and therefore it's going to be able to pass through a gap or a small hole or a slit, or whatever you want to call it. Meanwhile, starch is going to find it harder to pass through. So if you're given a question where you're asked which of the following would be able to pass through easier, would find it easier to pass through, or be able to diffuse easily through a membrane, you should know that glucose would be the answer to that question. Of course, I'm not going to ask you such a question very directly like what I've just said. So if you're given a figure, you should be able to know that smaller molecules will be able to move through a permeable membrane than larger molecules. Larger molecules would find it harder to pass through a certain membrane that is less than their size. It's a very important concept I've explained to you right now. The other question, or the other concept that I said I'll explain, is osmosis. And osmosis is a type of diffusion, but it is not exactly diffusion, right? So it is actually the movement of water through a semi-permeable membrane. Now you need to be able to distinguish between osmosis and diffusion. Osmosis is a diffusion of water through a selectively or a semi-permeable membrane. Now, in the case of osmosis, it's not solids or any other thing that's moving, it's only water that's moving through. So here's an example in this figure. In this figure here, we have 10% salt outside in the solution, and inside this cell, you want to call it, we have 20% salt. So we have 20% salt inside and we have 10% salt outside. That means that outside we have 90% water. And now how do you find the 90? It's 100 minus 10. And inside the cell we have 80% water. So if you compare both of them, the question you want to ask yourself is which one has the most amount of water, which one has more water? And it's very obvious that it's inside of the cell. So because the cell has, I apologize, it's outside of the cell. And so because outside of the cell has 90% water and inside the cell is 80% water, 90 is greater than 80%. So therefore, in osmosis, which is a type of diffusion, we said that water would move from a region of high concentration, which is 90% outside, to a region of low concentration, which is 80% inside. So by osmosis, water is going to move into the cell, right? So water moves into the cell. And that's a very important concept that you need to know for the whole questions I'll be giving to you on your test. So I'm going to repeat very quickly that osmosis is the diffusion or the movement of water from a region of high concentration to a region of low concentration. Now, what is a hypotonic or hypertonic? A hypotonic solution is a solution that is the solution that has the most amount of water, or the solution that has more water. So in our case here, this outside of the cell has 90% water, therefore it's the hypotonic solution. Inside the cell has less water, it has 80% water compared to 90%, so therefore it is hypertonic. Okay, that's something you need to know. Water travels down its concentration gradient from high to low. Basically, what this is saying is that water always moves from a region of high concentration, as where there is more water, to where there is less water. And in osmosis, it doesn't require energy, right? So it just happens passively. It happens without you forcing it to happen. This process would continue to happen until we establish equilibrium, until there's equal amount of water in both sides, both inside the cell and outside the cell. And because of this concept of equilibrium, we looked into how this could cause a cell to rupture or to burst. If I place a cell in an environment that has so much more water than inside of the cell, the tendency is that if the cell is an animal cell, too much water is going to go into the cell, and that could cause the cell to rupture or to burst, like you see in this figure. So that's something that we don't like for human cells or for animal cells. But for plant cells are fine with it. Plant cells have a cell wall, and because of the presence of the cell wall, they like when they take in so much water because it makes sure that the cells are there's so much more pressure inside, and that gives the plant a very strong stem or branch or whatever structure that it needs to have to function properly. All right, so the opposite is the case. When you place an animal cell in an environment that has less water than the inside of the cell, it means that the cell is going to lose some of its water to the external environment. And that's not a good thing as well, because we want to make sure that there's a balance between the inside and the outside. So in this case, when the cell loses its water, it's going to shrink, right? The same thing happens in plant cells. So this is not a good thing, and this can cause cells to die. Right, so here are different examples, or here's a summary of all of these things. In an isotonic environment, there's equal amount of water inside and outside. It means that even though water moves in and out, there's going to be a balance. So at the end, there will be no net movement of water. In a hypotonic solution, which is this gray solution, there's so much more water, and therefore water is going to move into the cell, and that could cause the cell to rupture or to die. In a hypertonic environmental solution, you place a cell in a solution that has very little water, and therefore the cell itself is going to sacrifice its water to the external environment, and that would also cause the cell to die by shrinking. Here is, in terms of numbers again, inside the cell we have 35 solute and 65 water, 35 solid outside and 65 water. So what kind of cell is this? What do you think is going to happen? Obviously, it's an isotonic environment, and the reason why it's isotonic is because it has 65% water inside and 65% water outside. So therefore, there's going to be a net balance of water moving in and out. All right, so that's it for this topic. If you have any questions, please do shoot me an email or ask on Google Classroom. Thank you.